All right, so we'll get started. It's 11 o'clock. Welcome to the fall 2023 GES Symposium. We have 3 students today presenting their faculty mentor research results. The format goes with the students. They'll present their work. Actually, first the mentor will come up and do a short introduction. Then the student will present their work. Then we'll take questions from the audience, or the student will field questions from the audience. At the conclusion of all our presentations, there'll be food and post 121. We do record these, so please do not do any movement. I guess this has some sort of voice or visual tracking, so please only get up to go if in between the talks. And then finally, two things before we get going, make sure to silence your cell phones. And then also I'd like to thank the GES support staff, Tina, the rest of the coaching office, Kristen, Alpha, on Maria, and then obviously all the Oceanography faculty for the support program, and the faculty continue to step up across campus and support our students. So our first speaker is Nadia Einfalt, and it's on deck.
Good morning, everybody. I had no idea that I would be doing this, so I'm totally happy with it. Nadia has done a great job studying the impacts of sea level rise on the remote island of Yap. It was very difficult finding a data set, a digital elevation model that we could use for mapping the impacts of sea level rise. But she persevered and did a great job. Sea level rise, many people don't really realize that it is an inevitable, accelerating, and unstoppable reality in our lives. The Intergovernmental Panel on Climate Change Assessment Report number 6 states that because of ice sheet melting and deep ocean heating, sea level rise will continue for centuries to millennia. So this means that every coastal community in the world, and especially low-lying islands like Yap, need to really change their game. We need new policies, new laws, and we need to figure out what to do with all the infrastructure that's already located along the shoreline. And so studies like Nadia's are exactly what we need to begin the process. And the last thing I'll say is that Yap is one of the 4 states in the Federated States of Micronesia. These are severely under-resourced nations that have traditionally and historically lived close to the land, but because of colonialism and the influence of our global economic models around the world, which are so extractive, they are now very dependent on shipped-in food, medical, construction materials. Their economy really is dependent on external sources. And so one way to look at the challenge of sea level rise is it's an opportunity for us to change our models, to get back to closer living to the land and building resiliency in the process. So Nadia, take it away.
Thank you for that introduction. Hello, everyone. My name is Nadia Einfalt, and today I will be talking about sea level rise impacts and adaptation on Yap proper. First, I'm going to begin with a little bit of background on sea level rise and Yap proper and its people. As Chip explained very well to us, sea level rise is a continuing phenomenon that is a result of higher atmospheric and oceanic temperatures as a result of greenhouse gas emissions. It is primarily a result of 3 mechanisms, the first being thermal expansion. Thermal expansion is where the oceans absorb atmospheric heat, leading to an expanding water column. and this is responsible for about 46% of global mean sea level rise. The second mechanism is melting continental ice sheets and glaciers, which is a result of increased atmospheric and oceanic temperatures, and is responsible for about 43% of global mean sea level rise. Lastly, we have the persistent anthropogenic extraction of groundwater for agriculture and industry, and this mechanism is responsible for about 11% of global mean sea level rise. Sea level rise can have many impacts on a community. One of the impacts is more frequent and severe flooding, and this can include sunny day flooding, where high tides are paired with high sea levels and results in flooding on days with no rain. Another impact of sea level rise is coastal erosion. Increased rates of coastal erosion are happening on beaches all over the world, and as you can see, it's also happening in Hawaii. That is Sunset Beach on the North Shore, where houses are falling into the ocean as a result of increased erosion rates. And lastly, we have saltwater intrusion, where higher sea levels force saltwater through coastal wells and aquifers, leading to the contamination of potable water resources. And this is particularly alarming in communities that are already water stressed. So now I'm going to tell you a little bit about Yap. Yap is one of the 4 federated states of Micronesia and is composed of 135 islands, 22 of which are inhabited. Of the 22 inhabited islands, there are 4 main islands that are conjoined and make up the main island of Yap, also known as Yap proper. And the population of the state is about 11,300 people. The people on Yap are already feeling the effects of sea level rise. Typhoons from 2002 to 2013 have resulted in severe flooding. This flooding led to the loss and damage of many coastal assets, including roads, markets, and ports, which can severely impact economic and food security. Additionally, they're also experiencing the salinization of crops. Severe flooding has led to the salinization of almost all crops in low-lying areas. And additionally, a 2016 study found that 20% of households that have land for agriculture on Yap have experienced damage due to salinization. And this is the highest of all of the federated states of Micronesia. Their cultural assets are also at risk due to sea level rise. Traditional men's houses, called falu, which can be seen on the screen, have been repeatedly rebuilt because of damage due to erosion. Additionally, members of the community have stated that these houses have been continuously flooded more frequently and severely in recent years. Their traditional stone money, called rai, also seen on the screen, have been also impacted by sea level rise. They have been lost to severe floods and also experienced dissolution and corrosion as a result of continued exposure to saltwater. These problems are also paired with the other challenges on Yap. Firstly, they are disproportionately affected by climate change. Small island nations only emit about 0.2% of global greenhouse gas emissions, yet they feel some of the worst impacts of climate change. This is in addition to the fact that their under-resourced areas have a lack of localized climate data, have a lack of elevation data, and no localized erosion rate data, which means it's very hard to identify and plan for future sea level rise impacts. This brings me to my project. The goal of my project was to better understand how 1 meter of sea level rise will affect the communities on Yap through the use of geospatial modeling. To do this, I utilized ArcGIS Pro, which is a geographical information system software that is a powerful tool used for mapping and spatial analysis. I created a hydrostatic flood model within ArcGIS Pro that showed what 1 meter of sea level rise would look like on Yap proper. The data I used for this project was a digital elevation model, or a DEM, and several asset layers that showed the locations of important resources on the island. After processing the data, I was able to use the spatial analysis tools within ArcGIS to better understand exactly what assets would be inundated under 1 meter of sea level rise. This is the DEM that I used in my project, and it was created from commercial satellite imagery from Vitron in 2018, and it has a 0.5 meter resolution, an absolute accuracy of 3 meters, and a relative accuracy of 1 meter. Now we're going to go on to the results of the flood model. This video shows the before and after of the flood model, with the blue areas depicting areas that will be flooded under 1 meter of sea level rise. I zoomed up on this particular area of Yap because it is Colonia, the state capital, and the main center of commerce on the island. Now we can go through the results of the model sector by sector, starting with businesses and other structures. Of this sector, about 2.4% were inundated, which was about 100 out of 4,000 buildings or other structures on the island. And the inundation of this layer did occur island-wide, but did have a concentration in Colonia. Next, we have the government and NGO asset layer, and this asset layer had an inundation of about 5%, which was about 10 out of 191 total facilities. And the inundation of this layer was also highly concentrated in Colonia. The inundation of this layer included the sole public library on the island, the National Police Office, and the Gap Living History Museum, which offers live demonstrations of traditional skills to the community. Now we can move on to businesses. Of this layer, about 34% was inundated, and of the 16 inundated assets, 15 were located in Colonia. The inundation of this sector included essential businesses such as a car mechanic and a gas station. It also included several hotels and restaurants that support the local dining, tourism, and recreation, and other hotels and coastal businesses that also support the economic security of the community. Next, we can move on to the critical infrastructure, and of this layer, about 36% were inundated. And this inundation was also highly concentrated in Colonia. The inundation of this layer included several wastewater facility sites, the FSM Petroleum Corporation, which supplies the island with fuel, and one of only 2 ports on the island. There were also many buried infrastructure that will be impacted under 1 meter of sea level rise, including about 1.27 kilometers of water line, about 1 kilometer of sewer line, and a little over 6 kilometers of roads. The inundation of critical assets is particularly alarming because one of only 2 ports are inundated, and they obtain many of their resources from importation. Also, the inundation of wastewater facilities can lead to the contamination of potable water resources in coastal waters. Now we can move on to the inundation of cultural sites, where about half of all cultural sites on the island will be inundated under 1 meter of sea level rise. This includes 23 fallow and 4 chabog, 1 dancing ground, and 1 church. As you may have been able to tell through the past few slides, the most impacted area under 1 meter of sea level rise will be Colonia, which is the main center of commerce for the island and the capital of the entire state. This could suggest that it could be a good potential site for future adaptation plans. Like any other project, mine too had some limitations. The first being that I used a hydrostatic flood model, which does not include the dynamic elements of water. And it also does not include coastal erosion and groundwater inundation, which could lead to an underestimation of the total extent of flooding for 1 meter of sea level rise. Furthermore, I have no firsthand experience with this area, meaning that I was not able to ground truth the data, and I had to use predetermined asset layers, which sometimes lacked the detail that I would have liked. So now that we know about how sea level rise will impact the community, what will we do? In the face of sea level rise, many communities look at adaptation. And it's important to acknowledge that adaptation has been successfully practiced by Indigenous communities for thousands of years. So in order to build truly comprehensive adaptation plans, it will be integral to pair this traditional Indigenous knowledge with modern climate science. Adaptation can commonly be broken down into several categories, and today we'll be talking about protection, accommodation, and retreat. Protection refers to building defensive mechanisms in order to safeguard the community against the impacts of sea level rise. One way to do this is through hard protection, such as dikes, groins, breakwaters, and seawalls. However, hard protection measures commonly have negative consequences, such as high cost and maladaptive consequences. There are also soft protection measures, also known as ecosystem-based adaptation, that can be used. One of these adaptation measures is revegetation of the coastline. One way to do so is using mangroves. Mangroves are native to Yap, and revegetation efforts are being used today. Furthermore, beach nourishment and sand dune restoration can be used as a soft protection measure to create a line of defense between the oceans and the coastal communities. Next, we have accommodation, which is modifying existing structures to withstand the effects of sea level rise. One way in which to do this is to elevate houses or roads, so that way flooding moves underneath the facilities. Another way to accommodate is also to use different agricultural techniques, such as climate-smart agriculture or cement taro farming, which is already being used in Yap, where taro is grown in cement planters in areas that are becoming increasingly saline. Next, we have retreat, which means moving coastal resources away from the coastline to protect them from the effects of sea level rise. And this is best used in a long-term adaptation plan with phased implementation that have specific trigger points. I would like to thank the Climate Resilience Collaborative, namely Chip Fletcher and Juliet Budge, for helping me through the duration of my project and research. And I would also like to thank Vikron for supplying me with the data used in this study.
Questions for Nadia?
I'm sorry. Yes. I haven't been down to Yap, so that was kind of one of the limitations in my study, is that I wasn't able to actually interact with the people there. But that's something that I think would be really great for future research.
Can this be done everywhere in the world?
Yeah, so flood mapping can be done anywhere where you have elevation data. So as long as you have a digital elevation model, you can do flood mapping. We do have it here for Hawaii, and I also believe they have it for the continental US through the NOAA Sea Level Rise Viewer.
No, but the question is not whether people can do it small scale in other places. The question is whether can it be done everywhere, this thing. Right now, can we do it?
Oh, can it be done everywhere in the world? Not so places that are kind of under resourced as far as climate data and don't have those elevation products would find it more difficult to make some flood models. Yeah.
You mentioned this scenario of 1 meter of sea level rise. Do you have an estimate of what sort of timescale that would occur?
Yes, so if you look at the NASA sea level rise projections, it shows about 1 meter of sea level rise for this area in year 2110. And that is for the 3 higher scenarios.
When you talk about sea level rise, what's the definition you use? Do you do 1 meter increase and stay there, or is it like the chronic flooding at 1 meter?
So this is just a hydrostatic flood model, so it's just raising it kind of like a bathtub, like across the lake.
Question was asked.
Okay. Any other questions?
What made you pick Yap?
So that's a really great question. I think one of the reasons was that Micronesia specifically is such an under-resourced area and they didn't have any flood models. So it felt like even with a simpler model such as a hydrostatic model, it would still be a useful thing to do there, whereas other areas already have more dynamic models. So doing something like this for those areas wouldn't be as useful.
Are there other areas that are lower than sea level than Yap or something like that?
Yeah, so Yap does have a little bit of higher elevation land within it because it is a high island, but many of the atoll islands throughout the Pacific are lower in elevation, and they're experiencing even worse impacts of sea level rise, even sometimes complete migration from those areas.
How are you making your model available to the people on the island of Yap to actually look at and be able to utilize?
Yes, so through part of my project, I worked with a woman named Maria Cotton Meyer, and she works for the Digital Atlas of Micronesia, which is where I downloaded all of those asset layers from. And the digital Atlas of Micronesia is a really great online space where these geospatial layers exist, and she was really interested in maybe integrating it as part of that program. And she's very connected within those communities to kind of spread it out as well.
Our next speaker is Keanu, Rochette-Yu, Tsuen. Over to the Alegado.
Yorana and aloha everybody. I want to greet you in Keanu's native language of Tahitian as well as Hawaiian. I've known Keanu since he was actually an undergraduate at Kokilani Community College when we were introduced and he wanted to possibly join the GES program and became a part of the Miami Mentoring Bridge. Once he transferred over, he was still really interested in research and we brought him into the Native Hawaiian Science and Engineering Mentoring Program where he worked with my graduate student, Eric Tong, and that's where he first learned that there was community level research happening in Tahiti, which is where he was from, and he had never heard of that before. For that short period of time in my lab, he learned molecular techniques, which he applied in his project, which you'll hear about today. And I think I just wanted to bring up that story that while Keanu went on to do really great research at HIMB, at the Smithsonian Tropical Institute, you can read about all the places in here, and went on to be awarded the Hollings and all kinds of really, really wonderful funding opportunities. I think that seed was planted and he wanted to come back for his GES thesis and work on problems that were really important to local communities, local Indigenous Pacific Islander communities. And so for his project, he really wanted to support the practitioners of Hawaiʻi Fishpond. And as for those of you who have volunteered, we get in the water and we get dirty, but we wanted to know what is the risk of those of us who might be volunteers and those who are practitioners to some of the pathogens that might also be the result of land-based pollution. And that's the study that Keanu has taken on and will be presenting today. So we're very proud.
Thank you, Rosie, for your wonderful introduction. I couldn't have worded it better myself. Share that with the people. Well, Yorana, everyone, hi, my name is Keanu. Like Rosie mentioned, I am from the island of Tahiti, yes. And today I will be presenting my GES thesis, which is about analyzing different environmental factors that may correlate with the presence of a family of bacteria, the family Leptospiraceae, in the Hawaiʻi fish pond. So this project is centered around the disease leptospirosis. It is a zoonotic disease that is caused by a bacteria in the Leptospiraceae genus, and is typically transmitted to humans via contaminated water from the urines of mammal carriers. And these mammal carriers for Hawaiʻi are specifically feral swines. And so the disease itself manifests itself in a various array of ways. It can be very mild with fevers and chills, and can get more severe with jaundice, diarrhea, vomiting. And in its most severe form, it can cause multiple organ failure, such as liver failure, kidney failure, and potentially cause death. Most of the cases occur in the tropical band of the country of the world. So 73% of the cases. And when we look at the national statistics for the United States, Hawaii has the highest incidence of the disease in the whole country. And we're talking about the 100 times the national average, which is quite a lot. And this is partially attributed to a renewed interest in traditional farming. And there are some cases that are related to recreational activities, but tower farmers use wetlands in order to grow their crops. And so they're chronically potentially exposed to potentially contaminated water. So they're more at risk. This is our study site. This is the Ahupua'a of the area. And for those of you who are not familiar, an Ahupua'a is a traditional Hawaiian land division that contains complex agroecosystems. So site B represents the wetlands or the low ecolo where most of the crops are being grown. And this is the site where most of the farmers might get sick of leptospirosis. So this watershed, we know from local hunters that there are populations of pharaoh swines that live in the mountain. And our assumption is that as the water trickles through the watershed, it's going to pick up these bacteria and put them into this area of high population density. So the low is where people are working, and all the water is going to end up in the Hei'a Fishpond in Site C, which is our study site. And this site, we know that all the water coming there because that's the terminal point in that watershed. So based on literature, we know that leptospirosis outbreaks are correlated to different environmental factors. The one that we're going to be focusing on most on this presentation are going to be the rainfall and flooding aspects of it. I do want to have a disclaimer that as this project is part of a bigger project of community analysis, we are looking at a broader category of bacteria, which is the family of Leptospiraceae, not specifically Leptospira. And so from those, we developed 3 hypotheses that are going to help us study that bacterial distribution. So the first one is that we're going to find Leptospiraceae family more during the wet season of the Hawaiian Islands. The second one is that storm events are a major driver of Leptospiraceae input in Hei'a Fishpond. And the last one is related to the nature of the area. So Hei'a Fishpond is a constructed estuary, so there's a gradient of salinity throughout that fish pond. And so we're going to find these bacteria more during fresh water rather than salt water. So in order to validate our hypothesis, this is what we did. So my lab has done 2 sampling campaigns of water. The first one was in 2014 to 2015, and the second one from 2017 to 2019. So they collected water samples throughout the whole fish pond, as you can see with all these sample sites. And they also collected additional data such as nutrient salinity, and I collected precipitation data from online database. But we're going to be focusing mostly on the salinity aspect here and the precipitation. And then from the water samples that they collected, they extracted DNA from the water column and then amplified DNA and classified them in different taxonomic groups that we call amplicant sequence variant. So as an example, this is how we can cluster them. And as a reminder, we're going to be looking at the Leptospiraceae family here. That contains 3 different genera of bacteria: Leptospira, Leptonema, and Turneriella. As a reminder, Leptospira is the bacteria of interest that may cause leptospirosis, and not all of them in this group are necessarily pathogenic. So the first variable that we looked at is the seasonality of the distribution. So in these bar plots here, we have the average number of Leptospiraceae that we found in the fish pond by season and by year. So when we looked at these bar plots, we noticed that overall it seems like Leptospiraceae relative abundance is higher during the wet season. I do want to acknowledge a slight anomaly in our data set here, but it might be related to a non-typical rainfall year. And so overall, it is higher in the wet season, and we decided to test the strength of this association using an ANOVA and a Tukey's test, which is what you see in these box plots. So these box plots here have the 2 A's here, which signify that unfortunately, when we tested the differences, it was not significant statistically. That doesn't mean that it's not valid, it just means that it was not different enough that it was a jarring difference. But this difference is noticeable on these graphs. The second variable that we looked at is precipitation. So we have 2 linear regressions here that represent the 2014 time series and the 2017 time series. In the first time series here, we can see a nice positive trend with a strong R-squared number, which is our correlation coefficient. So it seems like precipitation is correlated with higher number of Leptospiraceae. But when we look at the 2017 time series, we can see that this R-squared is very small, close to 1. And that the pattern is not the same as in 2014. So we looked at the data, and what we notice is that these 2 points here are the ones that are mainly driving these regressions. And these correspond to very strong storm event with very, very heavy precipitation. So the 2017 actually is a very good pattern of baseline of precipitation. whereas the 2014 is what we would see during storm events. So we can see here that the Leptospiraceae relative abundance would be higher during storm events. And finally, the last variable that we looked at was salinity. So as we can see here, these are plots of the fish pond in both the wet season and dry season, as well as average salinity and average Leptospiraceae relative abundance. So the first thing to note is that the seasonal pattern that I was talking about earlier is visible in those maps. So when we look at the Leptospiraceae plot, there's much more red in the wet season than in the dry season. The second thing is that the salinity is a gradient across the fish pond. And in areas of low salinity, so in these lighter areas, we have more of the Leptospiraceae detected. So that suggests a potential correlation. And the third thing that I want to point out is that there is a stream that runs through the north side of the fish pond. And that might be an important factor in determining the distribution of these bacteria across the site. Yes, so they are more closer to the stream. So in order to test the strength of this correlation, we did the same thing, linear regressions. In the 2014 time series, we see a negative trend with a strong R-squared value. So it seems like the salinity is inversely correlated with the amount of bacteria that we detect. But in the 2017 time series, we see that our R-squared value is actually very, very small, close to zero, pretty much zero actually. And there's no correlation between the variables. So we wondered what happened in the span of these years. And we looked closer at the data and we noticed that in 2017, some of these sites are outside of the fish pond boundary. So they might be on the wall or outside. And the ones that are closer to the stream, like these sites here, are actually higher in variation of the bacteria. So we decided to take a different approach. Instead of just looking at salinity, add another variable, which is the distance to the stream. Yeah. So sites are important to look at. So this is exactly what we did. We measured the distance from the sample site to the freshwater input in the fish pond. And this is the linear regression that we got. So it seems that there is a negative trend again with a moderately good R-squared value. And that just shows that in addition to the salinity, what determines the amount of bacteria that are detected in this fish pond is the sample site. So there is some explanation that is due to the site location. So we have to account for that when we're doing our analysis. Now, again, we studied the family of Leptospiraceae. Not all of these members are pathogenic. So we want to know whether or not they represent a danger for human health and the workers in those fish ponds. So we use a technique called microbial source tracking in order to detect pathogen Leptospira in the fish pond. And this technique uses DNA markers in order to detect the presence and absence of a bacteria of interest. And so for us, we use the L32 gene as our marker for this bacteria. And we know that it's only found in pathogenic strains of the bacteria and it's conserved throughout. So we have 3 different tools in our tool set to detect this gene. And we have a set of primers, 2 primers, and 1 probe. And the way it works is that among the samples, we have samples with lots of DNA in it. And we're going to put all these 3 tools inside. And if one of the sequences is containing the gene, the primers and the probe are going to bind to the DNA. And so the primers initiate DNA replication, and the probe is in the way of that replication. So as the enzymes are working through the DNA, it's going to break down the probe, and the probe is going to release a dye that's going to fluoresce in the sample. So this dye is important because, one, if it fluoresces, it's an indication of a positive, so the presence of Leptospira. But the intensity of the fluorescence also gives us indication of the amount of DNA that is positive to Leptospira. So this is the result. We have our amplification plot. The machine sets a threshold detection here at 12,400, and all the samples across that threshold are considered positive. So we can see here that a lot of amplification occurred below the detection. And when we looked at our results, we noticed that the only lines that crossed that threshold were actually the controls and the standards, which we have put DNA in it. So all the samples that we tested did not amplify, so they are negative to Leptospira. And in order to validate whether or not this negative was a true negative, we still run our qPCR standard curves, which is what you see here. And we know how much DNA are in these standards, and we know where they're supposed to amplify. So the correlation between the data points are supposed to be very high, which is what we see here with our R-squared value. So that validates that our qPCR went correctly. And so the negative that we got from the samples are a true negative. So to wrap it up, this is again a reminder of all the hypotheses that I studied. The first one is that we're going to detect Leptospiraceae more in the wet season. So this one was validated. Although the results were not significant, we did see a difference between the season. The second one is when we compare the two time series of 2014 and 2017, we did see a difference between a storm year and a non-storm year. So this second hypothesis could be validated. And the third hypothesis was that we're going to find this Leptospiraceae more in freshwater than saltwater. This is partially validated because we did see a correlation between the two variables, but site location is also something that we should be accounting for when we're doing our data analysis. And the conclusion that is most important for community partners is that there's no pathogen Leptospira that was detected in that fish pond. So they're at a relatively low risk of infection. And if we wanted to redo these experiments, we're going to be focusing more in the low east, where more of the farmers are actually exposed to contaminated waters. And in order to even increase our the accuracy of our results, we can also put another primer inside our qPCR to detect the reservoir, which is the pigs. So we're going to detect bacteria that are associated with pigs, the Bacteroidealis bacteria. So these are all the people who have participated in this project. So I would like to thank the Alegado lab first for their support, specifically Rosie Alegado, who was my mentor, and Nadia who has been super insightful in this project, as well as the NIH Fund in maximizing access to research careers. Dr. Vivek Nirkar, who was the PI for this program, as well as Dr. C. I think they're actually present in the Zoom, so hello. And then the Europe program also funded this project, which is really important, so thank you to them. And we would not be able to do that without our community partners, so Pai o He'eia and the He'eianer. So thank you everyone for tuning in, and I'm open for questions.
Great talk. Thank you very much. Excellent study. One of your early slides that showed bar graphs, 2014 to 2019.
Yes.
Sort of hinted at a increasing trend that time period. Do you think that's real?
So I cannot rule it as it is. It might seem like so, but we also have to account the fact that it was a relative count. So it depends really much on how much samples were available to us, how much of the DNA of other bacteria were in our samples. So I cannot say that there's an increasing trend for say, just to be conservative.
Thank you.
Yes.
I presume that like any other bacteria, these ones reproduce very fast.
Yes.
And if so, then you will expect huge variability on abundance.
Yes.
And that obviously is going to determine your sample size. To account for that on your study.
So we did not really account for bacterial growth because what we did is as soon as the water samples came out of the fish pond, they were filtered the day of and put into the freezer. So there's an assumption that between the travel time, there was no bacterial growth in the samples.
I'm not talking about the bacteria, I'm talking about the fact that you need hundreds of samples to capture the variability within the pond.
Yes.
Did you have enough samples?
So that's the thing, right? So we were working backwards and we cannot just take samples and know that they're going to be positive. So it depends on how much of them come out positive, and then we have our sample numbers. Now for the statistical significance, it might not be the best number of samples, but that's the only samples that were available to us at this time. Yes.
Yeah, this relationship with precipitation is pretty tricky because it's a very nonlinear thing, right?
Yes, it is.
So I'm wondering if you have enough data to actually look at, rather than look at, say, wet season and dry season, but look at the characteristics of the precipitation. If it's like raining a lot over one day as opposed to very little over the whole month. And maybe after a while, you just, the rainfall is actually flushing the system rather than providing
That's actually a very good point because we did encounter a lot of issues when looking at precipitation data. So that's one of the caveats of the study. Unfortunately, it's not perfect. For example, our rain gauge was not even like in the middle of the Fishpond. It was the closest rain gauge that we can find next to it. So there's no dedicated rain gauge that measure precipitation on site. There's also another variable that we cannot account for in this study is the time of flushing, because these bacteria again are supposed to come from the watershed, right? So they're trickling through and end up in the fish pond. But what we did is just take the samples and assume that the flushing had occurred already. So we're just really difficult and challenging to account for. And if you have suggestions, then we'll be glad to take it. Yeah.
Yeah, if I may, another unrelated question perhaps. So the fish pond was put there for a reason,
Yes.
and presumably people that did that knew all about this connection between the mountain and the reef.
Yes.
So do you know why this, I mean, is this fish pond there because of oceanic conditions or?
Yes, I'm actually really happy that you're mentioning that.
Okay.
So Hawaii's watersheds are actually very short. And so there's a lot of flushing of nutrients within the watershed, right? So there's two aspects in the area. There was, maybe I should plot the map. It's going to be easier to talk about it. So there's two aspects here. There's first the low-E columns, right? These are their primary sediment capture zones. So once all the water is flushing, they contain nutrients and sediments. And so the Hawaiians have put these wetlands, these low-E's, in order to slow down the flow of the water and allow all the sediments to deposit in the low-E's. And so that allows nutrients to be present and make an efficient agricultural area. And on the other side, they're also maximizing the rest of the runoffs of the nutrient when they're flushing inside the fish pond. So the input of these nutrients in the fish pond allow primary production and also the efficient growth of fish. So they are pretty much maximizing all the ecosystem services available to them in order to have sustainable food production that can feed extended amount of people. And that was documented. So that was really, really good observations from them. Thank you.
So a chat question.
Yes.
How do you plan to communicate your results to the community?
Okay, very good question. I already did actually. So we are working pretty closely with Hi-Fi in order for them to be aware of what we're doing, right? And so they can benefit from these results as well. So I presented my result on, I think it was Tuesday or Wednesday, and they were aware of what we found. They know what is happening, and they're very happy with the research that we're doing. Yes.
Hey guys, nice job.
Thank you.
So there's this phenomenon that people have been observing for a long time called population, when fresh water is salt water is all of a sudden
Yes.
become salty. A lot of the organic matter will come together and stick out.
Yes.
So I'm curious if that thought about this process as it relates to your data set.
So it doesn't really relate to, well, partially. So flocculation, right, is when you have dissolved organics that become solid, I guess, somewhat when they encounter seawater. For us, we're not really looking at flocculation, but there is a dimension of the bacteria, the Leptospiraceae create biofilms and they aggregate to sediment particles. And so that's another thing that is important, is that since they're aggregating on these sediment particles, they may be sinking at the bottom when the sediments are going down, but relating to the low-E's, right? Because this is a sediment deposition area, the fact that the bacteria are clumping to sediments and they're falling down, that means that maybe, that's another caveat project, maybe all the bacteria have actually settled in the low-E's, and then the ones that have went through in the fish pond are non-existent. So that's actually a good point and relates to that partially.
It's very comprehensive.
Thank you.
Is there any data of cases of contracting leptospirosis from working in the fish pond?
Yes, from personal experience, I have.
It wasn't the fish pond.
It was not a fish pond, no. It was not a fish pond, it was the lobby. So contracting leptospirosis from fish pond, so from low E work, yes, there's documented cases. From working the fish pond, not yet, which is good.
Have you done any analysis of that data?
No, I was not able to because I focused mostly on the environmental data and not the clinical data. If there's no more question, thank you so much everyone for it.
So our last speaker is Nicholas Hawaiʻi GES and Camilo Mora.
So you're trying to say that's what you need.
Thank you.
So I was asked to do a brief introduction for Nick, and all this week I have been thinking about one word that would define Nick from the word that he has done for me. And before I tell you the word that I came up with, I'm gonna tell you what he did. So we are working on this issue of how climate change influences food. Turns out that we normally find very bad things that are happening, but in the case of food, there might be good things happening from climate change. One of them being that CO2 is the basis for plants to produce protein. So it has been used as an argument that climate change is actually a good thing. In addition to that, the plants can survive drought even better whenever there is higher CO2. So all of the elements suggest that climate change is actually a good thing for the plant. However, the story is a little bit more complex because of the fact that there are interactions between biomes. So they can endure the drought, right? But unfortunately, if it is too hot, they need to evapotranspire as well. So if there is no water, they have to transpire, things get complicated there. So there is this need for us to have these multifactorial experiments. Unfortunately, for you to answer those questions, you need to build robots. Every one of those robots costs about $1 million, and we need about 300 of them. So what it means is that we have to develop these things to make this happen, and we develop these robots here at UH. I'm hoping that you display very well those robots there, but those robots are made out of things that we buy from Home Depot, and every one of those robots costs about $100. We can get the things, but unfortunately, there is a challenge to make those things happen. Given the complexity, it becomes a lot of, there are problems every time with these robots. And with that being said, they managed to put that off. Getting those robots to work, to get data, for me, came down to that word that defines Nick, and that word is impressive. And Nick, with that, thank you for being part of my lab.
Well, thank you so much to Camilo for that amazing introduction. Aloha ka'ayaka o Nick na po pomaka'ios ko no kado maya o Alegado Learning Center. How's everyone? My name is Nick Pomaka'ios. My mentor is Dr. Camilo Mora. I'm here today to talk a bit about my research into the effects of climate change on plant growth. So first, just to introduce myself through place. My home lies within the Ili Kapakapa, the community of Ke'alekulu, the town of Kailua, the Ahupa of Kailua, the Moko Ko'ola Poko, the Mokopuni of Oahu, the Hawaiʻi of Hawaiʻi. I wanted to introduce myself through that, not necessarily because my research is directly place-based, because it isn't, but because it's my relationship with this place and love for this land that did directly inspire my work and really my entire academic journey. Moving on to my research, as I think you're probably all aware, both human society and terrestrial ecosystems are heavily reliant on plant communities, whether that's through food, economic resources, ecosystem services, even just recreation. Like both the ecosystems and human society really are reliant on our plant communities. And unfortunately, climate change is influencing a lot of the environmental factors that plants do depend on, such as rainfall and temperature. And moreover, it's these environmental factors. It's not just like, you don't just have to think about temperature. It's also the way the temperature interacts with water availability water availability, with CO2 concentration, with nutrients. All these factors combine in these complex interacting ways and make it pretty hard to predict how climate change might affect plants. This graph here, it's extremely simplified, but just to kind of illustrate the complexity of the factors here. You have things like water and temperature influencing the enzyme supply of a plant. Enzyme supply affects photosynthesis, which then can impact growth and leaf area, which is then feeding back into photosynthesis. So all these different biological and environmental factors that really have impact on the way plants grow. And my research, that's what I focus on. I'm curious about like how these, well, I focus on three factors: water availability, CO2 concentration, and temperature, and focus on how these factors combine and interact to influence the growth of plants. And specifically, I'm looking at how they affect plant mortality, plant growth rate, and the eventual nutrient content of those plants. So here's a slide where I get to brag about my numbers. For our research, we grew 350. Well, first of all, this is the Mora lab. Got these, I think it's the closest thing I'll ever be to being in a spaceship. Got these crazy growth chambers that are run partially by robots. They allow us to regulate temperature, water availability, light, CO2 concentration. We grew our 350 Mānoa lettuce plants within these 10 chambers. And within them, we actually have 50 different combinations of treatments, composing of 2 different CO2 treatments, one at just the ambient CO2 levels in the room, which, because it is indoors, it is affected by the number of people in the building, ranging from about 430 to 520 ppm per day. And we're comparing that with about 750 ppm, which is kind of the business as usual prediction for the year 2100. So we're still figuring that out. Then we looked at 5 temperatures, ranging at 4-degree intervals from 20 degrees to 36 degrees. And then 5 different water levels based on the maximum water holding capacity of the media, which was permeability ranging from 60% to 100%. And once we planted our plants in there, we left them for 21 days and then the results. So just to go on a brief photographic journey through my research. We started all our plants from seed in these rock wool cubes you see here, grew them for about 14 days and transplanted the whole cubes to positive vermiculite in the growth chambers. From there, they stayed there for about 14 days until some of the ones that survived grew into the plants you see there in the bottom right. Once they were fully grown, we cut the plants at the base, put them in plastic bags, and then dried them in a scientific oven to create these freaky looking lettuce chips you see there on the bottom right there. To get our dry biomass data, we then weighed the chips, and then for nutrient content, we ground the chips up to a fine powder, and then it went to the CTR Agricultural Diagnostic Service Center to be processed for nitrogen content. And then for mortality, I just kept an eye on the plants throughout the course of the study, and if they no longer had green, I considered them dead. On the topic of mortality, moving on to my results, probably the most striking result in regards to mortality was that there's a very strong relationship between temperature and mortality, with both of our 36-degree chambers, regardless of CO2 concentration. At 36 degrees, we had 100% mortality, though it should be noted in the elevated CO2 chamber, those plants did hang on for a lot longer. We also noticed that water availability, at least in regards to within the values we looked at, water availability was a bit less important, though it did have a bit of an impact for the ambient CO2 chambers. And just in general, there was lower mortality in the elevated CO2 chambers, though as you can see, that was generally eventually negated by increased temperature. And just to briefly talk about these graphs, here you've got temperature on the y-axis, water content on the z-axis, on the x-axis, and then the colors represent levels of mortality, with the dark colors showing where we had the highest mortality. We've also got the max and min values shown up there, with 100% mortality at 36 degrees for both types of chambers, and then the ambient CO2 dips to about 50% mortality at the lowest temperatures, and the elevated CO2 had only 10% mortality at the 20-degree chambers. Next, we're looking at biomass. This was personally for me the most interesting. Again, we noticed the definite temperature connection with biomass generally increasing as temperatures decrease. However, with the elevated CO2 chambers, we actually have the highest level of biomass at around 24 degrees Celsius, which is very different from the ambient chambers, which just decrease steadily with temperature. And from the ideal temperature could be well below 20 degrees, the lowest temperature we looked at. We also noticed in all the chambers, we have the highest level of biomass produced at 85 degrees, at 85% water capacity, not at the high or low end of this scale, kind of right in the middle. And as I mentioned, kind of the most interesting thing is that as well as the increasing biomass production, the elevating CO2 actually changed the optimal growth zone for lettuce. It actually made us that it does best at a temperature higher than modern day CO2 concentration. Moving on to nitrogen. Here the pattern wheel is a bit more complex, but first of all, I will just have a disclaimer that nitrogen is not what people look at when they study lettuce. Nitrogen is usually used as a proxy for protein content and don't really eat lettuce for the protein. However, protein is an important part of the nutritional value of any food, and it is commonly studied across a wide variety of plants, and it's easy to test for. So nitrogen was the nutrient I ended up looking at for this study. And what we noticed most strikingly, the nitrogen content had a pretty dramatic increase with increasing temperature, surprisingly, and also it was highest under high water concentrations. But we did notice that nitrogen content was actually a bit lower in the elevated CO2 chambers, which could indicate that as temperatures increase and CO2 concentrations increase, we might see a reduction in protein content in plants such as lettuce. So just kind of zooming out a bit for the big picture, kind of the takeaway for me was that increased CO2 associated with climate change does have the potential to promote plant growth and promote lettuce growth with the increase in biomass and reduction in mortality. However, eventually that does get negated by other environmental conditions such as temperature and water. As we found, like with water availability, mortality and somewhat mortality, but more biomass does decrease with both decreasing and increasing amounts of water. So both drought and flooding can have a negative impact on lettuce growth, and those are the two things that the two trends we expect to see with climate change. And also the heat waves and just generally higher temperatures are also going to have a negative impact on plant survival and growth. And also, even if we do have more biomass growing due to increased CO2, it could well be possible that the resulting biomass has less nutritional value for the people who eat it. And just to acknowledge some of the limitations of this study, because we did have such high mortality in a lot of the chambers, especially the ambient chambers, that did reduce the statistical significance of what we had. And also because of the way we controlled the ambient CO2, we just had it connected to the outdoors essentially. So it did fluctuate over the course of the day, which could impact the way the ambient plants grew. And with the experimental design, we studied with sterile media, with sterile vermiculi, which is inorganic, so it doesn't have a natural microbial community, which is something that is important for a lot of plants. All of the things we studied had uniform nutrient availability, and nutrients are not another thing that influence plant growth. Limited ranging conditions, we generally looked at like the upper levels of water availability and temperature. We didn't so much look at the lower temperatures and the lower water availability, so those could be valuable things to look at in future experiments. And we only looked at one cultivar. Different types of lettuce respond differently to environmental conditions. So looking just at Mānoa lettuce doesn't necessarily show us the whole picture of how lettuce will respond to climate change, but it's definitely a start. And all of these things could be explored in future research. So limitations, but also opportunities. So essentially, why does this matter? Why do we care? At the end of the day, we don't really know how plants are going to grow in 100 years. A lot of people have put a lot of great work into studying the way that temperature, that water availability, CO2 will influence plants. But if you don't combine all of those factors together, you're not getting the full picture. So we need more studies like this. We need more studies that combine the different factors that are going to encompass climate change and anthropogenic change in general. And we need to study that if we are going to get an idea of what our future holds. Because if we don't start researching now, we're not going to know what's coming, we won't be able to prepare for it. Some acknowledgements, I'd like to thank so much the Mora Lab, Dr. Camilo Mora, Kira Webster's here today was huge help to me, Kyle McDowell was the technological wizard who kept the lab running. I'd also send a huge thank you to the GES program and SOAS, Heather Seidel, Tina Vila, Dr. Gudry, Mark Hansen, I don't think he's here today, but wouldn't have been possible without all of you, so thank you so much. I'd like to thank Dr. Kent Kobayashi of the TPSS department for both helping me design my experiment and reviewing my thesis. I'd like to thank Dr. Seth Kushinsky of the Oceanography department for reviewing my presentation. I'd like to thank the Department of Geography and Environment for hosting the Mora Lab, which I did all my research. The CTAR, you know, the CTAR, I can only do something words. CTAR for helping me to process my data, and finally the Europe program for providing me with the funds to pay for those testing. And that's all I have. Do we have any questions?
Great study, Nick, and really great job presenting your results. I've seen papers where cereals, wheat, rice, soy, all suffer the loss of micronutrients, zinc, vitamin B complex, and also protein, loss of protein, which is replaced by carbohydrates, and higher levels of CO2. And in fact, there are projections that this effect will lead to severe problems with malnutrition in human communities. So how would you steer your research to look at the findings that are published in those studies?
You mean like in regards to how?
Well, your research suggests a beneficial result of raising CO2, and yet here are some studies that indicate detrimental effects.
Yeah, that's the thing, because just because there's more biomass doesn't necessarily mean it's doing the same amount of good for you. And in the case of our study, we did find the kind of a negative correlation between increasing CO2 and decreasing nitrogen content. So I think if we're to look at this more in the future, I'll probably look at other micronutrients like you're talking about, to see like if lettuce is losing, like sure lettuce is gaining biomass, but if it's also losing nutrients, losing protein, losing other important compounds, like is it actually beneficial or is it just leading promoting malnutrition?
Great, thank you. Yeah.
I had a question about your temperature control. Is that set to be constant all the time, or do you try to simulate the day-night difference? Because I'm wondering if that, climate studies are showing that this day-night difference as being.
Yes, great question. Actually, I'll show the diagram of the lab. The way the lab works, we have a.
Now we get to see the robot.
Yeah. Oh yeah. So this is the robot. It runs down the line lifting pots. The job of the robot is to control water level. Sorry, this is a tangent, but since you mentioned the robot.
Yeah, no, go for it.
Yeah, the robot runs down the line, it lifts up the pots and it weighs them, and then to regulate the soil moisture, it waters it to a predetermined level until a certain weight, and by weight we determine the water content of the soil. And in regards to temperature, we have a heating cooler unit in there that will basically keep a constant temperature during day or night. However, day and night were different degrees, different temperatures. I believe our nighttime temperature was 10 degrees or 5 degrees lower than daytime temperature. So it was not constant throughout the day, and it was on a 12-hour on, 12-hour off cycle per day.
Thanks.
Yes.
You sterilize your media, so you just autoclave it. How much it interacted?
The media actually comes sterile because it's inorganic vermiculite, so there's not really much for microbes to feed on there, so it was sterile.
So did you guys look at other work done in the field without simply getting soil?
Yeah, actually, we're looking at just previous research in this lab setting, this specific lab, and using stuff like peat moss or potting soil in the past did tend to lead to rotting and oxia, things that were not good for plants. So we use for
So scaling up, so like how could this be scaled up? Do you have like all these robots that go chamber?
well, that's the thing that Dr. Moore's design. It's like the whole idea behind this is that it's effective, but it's also affordable. And but the media vermiculite, it's not as cheap as say like peat, but like it's also not the most expensive media out there. Like, and there were suggestions like, okay, maybe you can add in like things like activated carbon, you can add in like special slow release nutrient, but what I kind of my focus on this was to both be accurate and to be cost effective. And I also had limited funds available, but we made it work.
Question in the chat. Do you have any comment on soil impact considering changes in the elemental content of plants in an elevated CO2 environment? i.e. an increased need for fallow periods?
Yeah, yeah, there's, wait, sorry, clarify that question is asking about the needs of plants or,
I guess a comment on the soil. Plants are doing. How their composition is changing. What do you see? So if they're uptaking more nutrients, for example, then you might not have a longer fallow period. Assume they're getting.
nice. Yeah, that's not, we didn't look at that directly, but just based on the fact that we did notice differences in the composition of the plants, like, actually, I should have clarified for the when we're looking at nitrogen, actually, what we're looking at is percentage of the dry biomass of the plant. So like the difference in even just like 5% nitrogen, like 5% of the protein in the dry biomass, not like after taking out all the water, like that is significant. So it does suggest that at least in terms of nitrogen, the plants are taking different things from the environment.
Yeah, they followed up to the soil quality perhaps decrease over time.
Yeah,
That's kind of what you get at, even though you weren't using soil, so you could test that.
yeah, no, it's definitely possible that as like plant needs change and what plants are targeting for as they grow, that could have negative consequences on the soil.
Why do you have a speculation on why you said you had a high mortality rate?
Well, definitely. I mean, like I said, we were definitely looking at the upper end of the temperature, which I personally think that's the single biggest factor for the mortality, because like Mānoa lettuce, like 36 degrees is a lot hotter than you'll see in any area where you're actually growing lettuce in Hawaii. So I think temperature is probably the major factor. Also, in the wetter chambers, like in the wetter pots, like because there was a lot of like stagnant, well, not stagnant water, but the soil was waterlogged, so that also leads to high mortality. Light could have a factor, because like we did use grow lights in there that I don't know exactly how Mānoa lettuce is typically grown commercially, but it could be that the grow lights are more intense than what they're used to growing in a farm. Yeah, those are the most. It also could be that like the nutrient content I put in wasn't right. I just kind of eyeballed it. And I did do some research on the nutrient content, but it was very approximate. So I might not have put in the right amount of nutrients, given that the media was sterile and had zero nutrients to begin with. So it could also be a factor. But I don't know for sure.
Okay, so you're saying that lettuce, which is like monocot plant, like grassy, what would be the difference that you would expect to see in other plants that are like dicots and have secondary growth, like trees?
Yeah, I believe that, well, I think lettuce is a dicot technically, but you're definitely right in that the way that monocots and dicots are different plant families, the way they're gonna respond to climate change in different ways. One major way is that I believe a lot of grasses, like corn, the way they uptake CO2 is on a day-night basis. So like they could interact with changing temperatures, depending on how climate change changes the temperature over like a daily scale. Also increasing CO2 levels might affect different types of plants differently, based on the type of photosynthesis they utilize. But yeah, essentially, like the responses to climate change very much are species specific. Like I'm not saying just because this one type of leafy green I studied did somewhat well under elevated CO2 conditions, that does not mean that every plant in the world is going to benefit. It's species specific, it's family specific. That's why there's a need for more studies on more plants. Yes.
Question is, what was the personal driver and motivation on this project? What made you choose it?
Few different factors. Actually, well, a little bit into the backstory of my research. I initially wanted to study this with guinea grass because I was concerned about the ways that invasive species are going to affect these islands differently under climate change. That eventually didn't work out for reasons I don't want to go into. But I then went from that into lettuce because, well, number one, lettuce to grow, but also because I'm concerned about like the way that climate change might affect agriculture, both on the large scale, like feeding a country, or on the small scale, just in Hawaii, feeding your community. Mānoa lettuce, it's a local crop, it's grown here, it does pretty well here. But I consider it as like, okay, it grows well here now, but what about 50 years from now? What about 100 years from now? Like, are we going to be able to be food sustainable with our current crops in the future? That was one of my research. Yes.
Yeah, sorry to go into over time, but it struck me that the system you built is super impressive. Your CO2 levels are still quite like high compared to like the seeds probably evolved in like a pre-industrial CO2 high 200s level. So I'm curious if like what would need to go into your system that you built if you wanted to be more precise about the CO2 control?
Yeah, okay, well, first of all, to be very clear, I did not build this. This is far beyond my engineering capacity. But the way that for our research, to control the CO2, the hard thing with lowering CO2 is that at least if you're using relatively affordable materials, it's pretty hard to keep like consistently lower the CO2 concentration in a chamber. So what we had to do is we just left a little gap in the wall of the chambers just so that the gas balances could equilibrate between the inside and the outside, which means that it is going to be, well, definitely higher than pre-industrial, but also even higher than just ambient over the globe because it's inside a building. The number of people in the building varies. Like even if I'm in the room for a long time, the CO2 concentrations start going up. So we just did the best of what we have, and that's part of why I chose like a rather high CO2 concentration for my elevator group, just so there's like a significant difference between the two.
Thank you. This concludes the symposium. Again, feel free to join the speakers, friends and family, post 121 down the hall here on right. Thank you again for coming.